Welcome to the New Jersey Mycological Association's Taxonomy Tuesday for May 21st. Tonight, we're very lucky to have with us Sam Bucciarelli, who is going to give us a presentation that looks through the lens of community science into the world of rust and other underobserved parasites of plants. Learn a bit about the complex ecology of rust fungi and which species can be found in our area. Sam is the president of the Philadelphia Mycological Club, yay, and an outspoken advocate for community science. She wears many hats, including educator, tour guide, and land steward, and I can vouch for her awesomeness in all three areas. She grew up in the Pinelands, swimming in tea water and picking wild blueberries. She brought her passion to all things wild to Philadelphia in 2017 and has been sharing this passion with others ever since. So with that, I'm going to turn it over to Sam for the evening. You're up, Sam.
Thanks, Mike. I'm going to go ahead and screen. All right. Can you guys?
We can. Or it's loading.
Perfect.
Now we can see it.
So I'm here to talk today primarily about rust fungi. We're also going to cover some other phytoparasitic fungi that are underrepresented on community science platforms, specifically iNaturalist. But we're Mycena leona.
So,
but then when I discovered,
Sam, we lost you after you said iNaturalist.
when I discovered rust. Oh, okay. Can you still hear me?
Yes.
Okay, cool. Let me know if you lose me again. I can try and maybe move closer to the Wi-Fi router if we're really having a hard time.
Okay, cool.
So my favorite color is orange, and I love anything that is orange. My favorite mushroom has always been Mycena leona, but when I discovered here on the screen,
so we lost you when you said when I discovered rusts.
Okay. Maybe let me move a little bit closer to the Wi-Fi router.
Okay. Take your time.
You know what? I'll turn my video off. Maybe that will help.
All right, cool.
Let me know if you lose me again. Sorry,
I
guys.
will. That's all right.
Cool. So on the screen right now, we have, on the left, we have Gymnoconia peckiana. I'll talk to you a little bit more about this one. But this is growing on a raspberry leaf, a blackberry leaf rather. And then on the right side, we have this beautiful photograph by Tom Bigelow. This is maybe one of my favorite photographs ever. And this is a rust fungus growing on a spring beauty flower. So Mike gave a wonderful introduction to me. And
so we lost you right after Mike gave a beautiful introduction to me.
Okay. Let me take a moment to move a little closer to my Wi-Fi router.
That's cool. Sure. Go ahead.
Would it help to call in over a telephone or mobile?
I think if she's got Wi-Fi, and can move closer to the Wi-Fi. That's the best option.
Agreed.
Okay. Hopefully
We can hear you now.
this is better.
Okay.
Cool. So Mike gave a wonderful introduction to me. Here's a picture of me laying on the forest floor where I can often be found, crawling around and looking
We
at all
can't
of
see
the
that
fungi.
picture. We're still on the title image, Plant Fungi Interactions.
Okay. Can you see it now?
Not yet.
Might be a stop share and then reshare. Okay,
There
there you
we
go.
can see you.
We got it.
Okay, great. About the technical difficulties. I'm on the forest floor. This is where I can often be found. I got into rust fungi because of an annual event that takes place in April, called the City Nature Challenge. And this is a worldwide event in which actually over 600 cities this year were competing and collaborating to see who could record the most biodiversity, and engage the most people in using the app iNaturalist over a 4-day period. So it always takes place the same weekend. It's right after Earth Day every year. It's the last weekend in April, which I'm sure you folks know is a notoriously bad time of year for mushrooms. But that is, of course, the thing that I fell completely in love with. And so I made it a goal of mine to continue to observe fungi during the City Nature Challenge, even if it was going to be challenging. What is out that time of year are plant pathogens, and perhaps the most charismatic of the plant pathogens would be their leaves. You
So
got
the
to
most
wonder,
charismatic would be,
the most charismatic fungi that are out at that time would be rust fungi. Are you guys able to see the violet on my screen now?
we can.
Okay, cool. So I ended up looking at a lot of things that looked like this. I would notice the purple violets, and take a closer look at the leaves. Quickly, through the power of iNaturalist, discovering that this is in fact a fungus. A closer look reveals so being orange being my favorite color, I quickly became very obsessed with rust fungi, and there is an incredible diversity of rust fungi in our area. Rust fungi are the second largest order of fungi, at least described fungi. The first being the agaricales, and so there's an incredible amount of diversity in rust fungi. They also have the largest genome of all of our described fungi. So we're looking at a huge number of species. We're going to cover just a small number of them today. If you're wondering where we're at with the fungal tree of life, here mushrooms over
So
here.
we lost you after you said, if you're wondering where we are in the fungal tree of life.
If you're wondering where we are in the fungal tree of life, we're over here in the Uridiniomycetes. That's this darker colored in area. And then this is pretty separate and pretty distantly related from some of the things that we would typically think of as mushrooms over here in the Hymenomycetes. So I have a video that hopefully will play for you guys. That's going to have a wonderful animation depicting the spore cycle of rust fungi. Most rust fungi are highly evolved. They are obligate phytoparasites, so they cannot exist without their host plant. And so because of the way that they have evolved, be species, and
Because of
a
the way they
threat
have evolved?
to can you guys hear me?
Yes.
Okay. Because of the way that they have evolved so closely with their host species, they do not usually kill their host species. I kind of like to use the analogy of like a mosquito. So a rust fungus might be kind of annoying to a plant that it's using as its host, but it's not going to usually kill that host. It can, however, become invasive in different parts of the world and may cause harm and maybe will kill the host species if it is brought to a different world, adapted to this type of pathogen.
If it's
And so
if
because
it's
it doesn't
brought
usually
to
kill
a
its
what?
host species, it's not
Uh-huh.
if it's brought to a different part of the world. So because
Got it.
because these fungi don't normally kill their host species, they are not typically something that receives a lot of funding for study, as I'm sure we know in this crowd. And most of the funding for studying rust fungi comes from agriculture. So we're going to cover the life cycle here of the wheat stem rust, which is a rust fungus that does threaten the food security of thousands of people around the world, maybe even millions of people around the world. And this is among the best. It is believed that rust fungi with 5 spore types, and then some species have evolved to only exhibit a few of those spore types. And we'll get into that a little bit later, but we're going to start with the with all 5 here with the wheat rust. So I'm going to play the video. Hopefully it plays. So we're zooming in to the spores. These are going to be Uridinea spores. The Uridinea are stage 2 of the spore cycle, but they are the only stage that can reinfect the same host plant over and over and over again. And so the Uridinea stage becomes extremely important, particularly to agricultural pests. This is what can lead to extreme of rust fungi that will decimate crop fields, producing these Uridinea spores. And they're going to fly away to new crop fields. They might fly away to other sides of the crop fields and continue to infect the wheat plants. The wheat stem rust is what we call a macrocyclic rust, which means that it will infect 2 different types of unrelated plants. So we're starting here on the wheat plant. We've infected a second plant now. As the colder temperatures move into the fall, the Uridinea spores are going to oh, we're still hybridizing in this animation. So this is an asexual stage of this fungus. So it will produce clonal spores of itself, but it does, because they are so prolific, occasionally mutate, and again, creating some really virulent strains of this rust fungus. So as the colder temperatures move in the fall, from the same structure, the rust fungus will begin to produce teleospores, which are often a much darker color and can overwinter on the plant. Not all transitions from Eurogeniospores to teleospores are triggered by the cold, but many of them are triggered by changes in weather. So we'll talk a little bit about that when we get over to some of the rusts that grow in this area. The teleospores will erupt and then begin to mature, sometimes forming a basidia, which is where we can do our gene recombination. This is a completely different in the genus Barber. That is you probably are familiar with Japanese barberry, which is this structure called a spermagonia, which is sort of like a pit full of hyphal cells. From here, it's going to produce an acia on the bottom of the leaf, the same leaf where it produced the spermagonia. And the acial stage is what I would probably refer to as the primary stage of a rust fungus. So this is the gametothallus. This is where we find the fungus to be highly evolved with the species that it produces its acial spores on. So this fungus, although it is known for infecting wheat, using the barberry plant as its primary host. We do find in this area, and that would be the cedar apple rust. So cedar apple rust, you may know, produces these very interesting structures that I lovingly call octopus balls. This is the teleal stage of this species, growing on a juniper. But the other host plant for this species is the gymnosporangium, and will infect
So
a juniper
we lost you
and a
when you
tree in the
said
Rosaceae
we're starting
family.
to talk about the other host species for this one.
Okay, the other host species is an apple tree for this specific species, but there are many fungi in the genus Gymnosporangium, all of which will infect a juniper tree as well as another plant in the Rosaceae family, usually a woody plant like an apple tree or a hawthorn or a serviceberry. All of those plants, the apple, the serviceberry, and the hawthorn, can be infected by this Gymnosporangium. And it would be important to look at the acial spores and the structure of the acia in order to determine which species it is that you're looking at. And that will grow on the leaf of an apple. So let's talk a little bit about the specific life cycle of cedar apple rust. And let's start at the acial stage, which is the stage number 1. So we will find the pycnidia on the fruits and on the leaves of the apple. And that is the structure that forms on the top of the leaf. And then we move to the acial stage, which is the structure that forms on the bottom of the leaf. This is the most important structure for identifying Gymnosporangium species. From the axial stage, we're going to skip the uridinyl stage in this particular species. This species does not produce uridinyl spores. The axial spores will move over to a juniper tree and begin to infect it, and then it will form a gall. It takes 2 entire years for the juniper and the fungus to form this gall together. From there, about 2 years into this process, the bright orange teleal horns will erupt from the mature gall and begin producing teleal spores. You'll see those teleal spores go from a bright orange to a darker, like more rusty color later in its life cycle. From there, the teleospores will go back to the beginning of the cycle with the pycnidia and the axiospores. So this is the life cycle of this particular rust fungus. This goes through 4 stages of spores. Not all of them go through all 5. This one has 4. Some of them will only have 1 spore stage, or will only be known from one of its spore stages. Let's talk about one that we find in the pine lands. This is the pine oak gall rust, Cronartium quersum. The genus Cronartium will always infect 2 and 3 needle pines as one of its hosts. Or Cronartium, this one specifically is oak, but other species in the genus
we're
Cronartium
still
can infect
seeing your cedar
any
apple
number.
rust page.
Okay.
There.
Okay, cool. So this is the Cronartium quersum. This is the axial stage growing on a pitch pine in the New Jersey pinelands. The genus Cronartium is always going to be infecting 2 and 3 needle pines. And then this specific species uses oak as its secondary host. But then other species in the genus Cronartium will infect any number of woody deciduous plants. So this is one that can be found extremely local to New Jersey. Again, this was photographed on pitch pine. The oak stage, I would say, is a lot more inconspicuous. We see these like dots and sort of wrinkles on the leaves of this oak tree. So it's not as easy to see on the oak tree. It's not as easy to notice it versus the axial stage growing on the pine, which actually deforms the whole branch or the whole trunk of the tree and then produces these bright orange structures. So these fungi look extremely different at different parts of their life cycle. Next, I would like to talk about Puccinia andropogonus. This is a rust that's growing on a beard tongue. So this is growing on the underside of a leaf of a beard tongue, which is a small herbaceous plant that produces these delightful white flowers on the top left. The alternate host for this species is a blue stem grass, which is how it got the epithet andropogonus. So we see it here. We see its secondary stage on the blue stem grass in the bottom right. So it is, again, a bit more inconspicuous on the grass. It sort of appears as these yellow or orange streaks or small pustules on the blades of grass. This species actually has a few other hosts as well. This species can be seen on Ohio buckeyes in addition to these 2 plants pictured here. It's worth telling you that the genus Puccinia is one of the largest genera of fungi in the kingdom. And so a lot of the things we talk about are going to be in this genus. But we've got a different genus now. We've got Coleosporium solidagonus. So this is a goldenrod rust. This is the underside of a goldenrod leaf that I photographed last year in New York City, actually right after leaving the New York Mycological Society Fungus Fest. So this uses goldenrod as its secondary host. So this is named after the second host. And then it uses 2 and 3 needle pines as its primary host, or its gametophallus host. The genus Coleosporium is always going to grow on pine trees, and then typically uses plants in the Asteraceae family as its secondary host. Next, we have ash rust. So this Puccinia sparagnoides is going to grow on ash trees. It most commonly forms on the petiole of the ash leaves or the ash leaflets. That's due to an invasive pest called the emerald ash borer. Given that this species uses the ash tree, its gametophallus host, primary host, we may be seeing a really serious decline in this species, and we may be wanting to consider this as a species that we may be losing at the same time that we lose our ash trees. However, we do have an abundance of dropseed grasses as the alternate host. Most of these fungi cannot continue their life cycle from their sporothallus host or their secondary host. So it is of concern that this species of fungus. We have the dogbane rust. This is Puccinia smilacis. So this grows on the underside of the dogbane leaves in its axial stage. And then it grows on greenbriers in the genus Smilax for its Eurygenia and teleal stages. I have seen it. I see it most commonly on dogbane, but I have seen it on Smilax in the New Jersey Pinelands while I was hiking the Baytona Trail last year. Oh, and then this is a fun one. So my friend Hannah Huber works as the Pennsylvania State Conservation Mycologist, which is possibly the coolest job title anyone has. And yesterday, this is Trangia prunis spinosae. Just to keep an eye out for this species. So it's photographed here growing on a ranunculus species. That's a buttercup flower. But we're on the lookout for this species growing on sharp-leafed hepaticas, which is a spring ephemeral wildflower. So she let me know that this species does grow, has been shown to grow in Pennsylvania in the historic records in the collections from hundreds of years ago, but has not been documented on iNaturalist in the state of Pennsylvania. So she was asking me to keep an eye out for this. I'm asking you to keep an eye out for this species. So it sort of on the undersides of the hepatica leaf, ranunculus leaves. And then it does also sort of give this almost camouflage appearance to the flowers as well. And then finally, it also deforms the hepatica leaves. So a typical sharp-lobed hepatica leaf is going to be a 3-pronged leaf, and it's going to be just a little bit fuzzy. But we can see on the left side of the screen here that this fungus has transformed these leaves to now have 5 points to them. And so this is one thing that you can look out for in the state of New Jersey. And if you're hiking in Pennsylvania, definitely keep an eye out for this species, Tranchelia pruni spinosae. So we'll be moving on from here to microcyclic rust. So everything that we've talked about prior to this point has been macrocyclic, having two different host species. From here, we're going to move on to microcyclic rust fungi, which only use one plant species as their host. And we'll start with the Mayapple rust, Alidus potuli. So this will grow on the underside of Mayapple leaves in the springtime. You will actually be able to see this from the top of the leaves. It will appear as sort of like yellow spotting on the leaves. But as this species matures, it is also going to turn a darker color as it begins to produce its teleospores. So you'll see it make the shift from this beautiful bright orange to a more deeper brown. And this is extremely common. I don't think I've ever seen a patch of May apples that did not have Mayapple rust growing on it. Next, we have the blackberry rust. So this is Gymnoconia peckiana. And this uses the Allegheny blackberry, the Pennsylvania blackberry, as its host. And it presents itself as sort of like an orange paint on the bottom side of the leaves. This can cause some damage to the plants. They definitely don't thrive when they have this rust fungus growing on them. And it does affect the production of blackberries. But I will say that if you are finding this as a recurring problem in your blackberry patch, you can plant red raspberries, and this species does not affect red raspberries. So if you are noticing that as a problem for you, you can just plant red raspberries instead. In the wild, out in our fields and meadows, this species doesn't seem to affect the population of blackberry health long term. And the Allegheny blackberry remains a staple of our transitional meadows going from meadow to shrubland. And we got the blackberries. Next we have the spring beauty rust. This is one of my favorites. Perhaps because this image by my friend Kevin really resembles an octopus tentacle, which really just does it for me. Of course, it grows on the spring beauty, which is another ephemeral wildflower that we see in the early spring. Again, this species does not appear to be causing major problems to the populations of spring beauties. It is just another thing that grows in a highly evolved way alongside the spring beauties. Oh, this is my favorites. This is the arrowhead rust. These calladii. Lands, which I did not find that day. But I noticed that this there was an orange coating on the bottom of this Peltandra leaf, and when I uploaded it, I did quite a bit of searching for this name. There had only been, I think, 13 observations of this species at that time, and I thought, wow, maybe this species is really rare. Maybe I'm onto something here, and I was really excited about it. And then I found this species on every single patch of Peltandra I found for the rest of the summer. So this is an extremely common species that is very underdocumented in our area, perhaps because it does grow in wetlands, which are areas where most people are not tromping around looking at the plants and the fungi there, especially the fungi. But this is very much present in our ecosystems despite being underdocumented. This is the groundsel crowned rust. So this is a species of rust that grows on an invasive plant, the golden groundsel. This plant is native to Australia. I would be willing to bet that this Puccinia is also native to Australia. I actually learned about this species while I was putting together this presentation. And as I was walking home from the coffee shop where I had been working on this presentation, I found it growing right outside of my house. So this is another extremely common species that grows all over the place. This plant, the golden groundsel, is extremely weedy. It pops up out of the sidewalks in the middle of the city, and this fungus is almost always present on it. So this is a really easy one to spot. However, because this is growing on an invasive species that is closely related to a native species, which would be our golden ragwort, Pekara aurea, there is concern that this could host jump to a related host and potentially cause some really serious problems for our populations of our golden ragworts. And we have not seen that happen yet. I suspect that this rust has been in the United States for a long time. But this is something that we should possibly be concerned about, especially if we are planting non-native species in our gardens. It is definitely a concern what we could be introducing. And so choose native plants. That's what I have to say about that. And here's the flower of the golden groundsel. Next, we have the multiflora rose rust. So this is Phragmidium rosa multifloridae, named after its host. This grows on the invasive multiflora rose, which is native to China. This rust is also native to China. When I first started studying rust fungi, maybe 3 years ago, this had not been documented in an official way as being present in the United States, but it is absolutely present here. It does grow on multiflora rose. There may be some concern that this could host jump over to our native roses, which would be really tragic because our native roses are not nearly as common in our landscape as this, as multiflora rose is. So that is this is another species of concern. Unfortunately, because this is highly evolved with the multiflora rose, it doesn't really cause very much damage to the plant. It doesn't, cause defoliation or deformation in any way that is a serious problem, which is rather unfortunate. And here's the beautiful flowers of multiflora rose. They're out. Another and will turn the tips of the leaves yellow. I've never found a Star of Bethlehem did not have this rust present on it. This one is maybe of less concern because we don't have too many plants related to the Star of Bethlehem that are native to our landscapes. But it is still interesting that this was essentially brought over with this plant and is now absolutely thriving anywhere where this plant can be found. So why would the rusts go through this to be phytoparasites, to do all of this host jumping and spore cycling and all of that complexity? Well, it is believed that since the rusts are highly potent, And
So we
then,
lost you after you said it's believed that since the rusts are highly co-evolved with their hosts.
oh, it is believed that since they are highly co-evolved with their host, that provides some stability to the population, and they can continue to thrive on that host. And then moving to the secondary host, the Sporothallus host, provides them with the opportunity to do some like gene recombination to bounce back. It is a way of having plasticity in the population to combat things like extinction events and maybe even, you know, climate change, which is unfortunately going to be an extinction event. So basically, all of this to say that the cycling that is performed by the rust fungi are to help it both retain its population numbers, and then also to further evolve new species. And I think that's pretty cool. So from here, we can talk about a few different plant phytoparasites that are present in our ecosystems that are, so we can have plenty of time for questions at the end. So I have the lesser celandine smut, Entyloma ficariae. This is present on lesser celandine all over the place. If you are looking at a lesser celandine patch and you notice that some of the leaves have this spotting on it, this is actually a fungus that is producing spores within these spots. If you're familiar with wheat lacoche, the corn smut fungus, maybe you've eaten that before, this would be related to that. And this is pretty distantly related to rust fungi, but they're often sort of grouped together because they are phytoparasites. This unfortunately doesn't cause significant damage to the lesser celandine as an invasive plant, but it is present in our ecosystems alongside of it. Next, we have the blueberry gall. This is fairly common in the New Jersey pinelands. I have found and photographed this species in Burlington County and Gloucester County. This is another smut that is growing a gall structure on the terminal buds of blueberry plants. But there are other species of Exobasidium that grow on rhododendrons and other ericaceous plants as well. So this is one that you should keep an eye out for if you are hiking the New Jersey pinelands. Next is my favorite. My favorite smut. This is the trout lily smut, Vonkia hofleri, and in the leaves of the trout lilies, which is one of our spring ephemeral wildflowers. I had been observing this species for probably about 3 years before coming up with a name for what I was looking at, although I did know that it was a fungus at the time that I was observing it. This is extremely underrepresented on iNaturalist. This is present on every single patch of trout lily that I have ever witnessed. Just keep an eye out for the bubbling of the leaves. I think that there are just a lot of people who are not keen on looking at this sort of thing, and it is also hard to document. You can see in the background image here that we've torn the leaf open and we sort of just see this like black smear. These are the spores of this fungus, and it would naturally rip open as the fungus and the spores matured. But yeah, this isn't exactly the cutest thing out in the forest, but it is here, it's present, and the data is not doing a good job representing that at this time. So I encourage you to put this on iNaturalist if you find it. We're moving on to ascomycetes now. I'm just going to go through a few ascomycetes that are under-observed but ever present. in our landscapes. So this is Alcinoe corny, the dogwood anthracnose. This presents as tiny little pink or mauve spots on the bracts of the dogwood flowers. So this is extremely common. It's on almost every flowering dogwood that I've had the pleasure to see. It is not causing major harm to this tree, although it may be causing, it may be opening the door for additional infections. It's not going to be what kills the dogwood for sure. The blueberry tar spot, Rhytisma vaccinii. This is another extremely common but underdocumented species. This will fruit in the fall, but can be found in the spring months in the leaf litter. Tar spot, I think, is a wonderful name for this fungus because it grows as sort of like a puffy, deep black bubbling on the leaves that is actually hard to the touch, much like a hypoxylon would be, but this is growing on a leaf. Next, we have the Dophistroma needle blight. So this grows on lots of different pines, particularly white pines, the eastern white pine. This presents as what you can find in the needle litter of almost every white pine tree in our area. Occasionally, if the tree is in an inappropriate location, or if it is in otherwise bad health, this will cause some yellowing of the needles and some defoliation in the pine trees. But typically, this species is not going to be what kills an eastern white pine, despite being ever present in the landscape. Next, we have the beech bark disease. This is a tiny pustule that was present on a small beech twig. I would say that this structure is about the size of a pen point. This is a disease that afflicts our beech trees and causes. So you'll notice some red streaking coming from these pustules on the trunks of the trees. You will also notice that occasionally the bark will change from a smooth silver to a more textured pattern. And then you will also notice a lot of root suckering happening in the beech trees. And so those are the symptoms of the beech bark disease. This is the fruiting structure of this fungus erupting out of a small twig. And then we have the cherry leaf curl. This is Taphrina farlowii. I see this often on young black cherry trees in our area. This is a fairly for how common it is. And it looks like this. It sort of is like this brown puffing or curling on the cherry leaves. Again, most commonly seen on young cherry trees. Oh, and then this one is a real treat. So this is the Andropogon false smut, Epicoccum andropogonus. This is a species that can be found overwintering on grasses in meadows. So for the first few years that I was observing this species, I assumed that it was a parasite on the grasses. It actually grows from the seeds of the grasses. So keep an eye out for it over the winter. But in fact, this is not a phytoparasite. This is actually a honeydew eater. So this is a mushroom, essentially, growing on the sticky substance that is being produced by a different fungus. And that would be the fungi that are in the Claviceptaceae family. Like ergot fungi that do infect grasses. So this is a secondary fungus that is eating the sticky substance produced by another fungus that is parasitic on a grass. This is extremely present in our ecosystems, and it's not incredibly underobserved on iNaturalist, although the data could be stronger. But I do want to encourage you to keep an eye out for this if you're walking through a meadow, particularly in the winter months. Here are a few things that are on my list of things I would like to find but have not yet found. So perhaps that you may want to find them as well. The moss rust, closest extant relative of rust fungi that grows as a phytoparasite on mosses.
We're still on the, oh, here we go. Now we can see it.
Okay, great. So that's the one all the way to the left. And it does present as these teeny tiny little almost coral structures growing out of the mosses. This should be at its peak in June and July, so I'm hoping to see that this summer. I will be on the lookout, and I want you to be on the lookout as well. We've got striped wintergreen rust. I've been eyeballing all of the striped wintergreens in the area. I have yet to find this one. But I want to encourage you to be on the lookout for it as well. Striped wintergreen is a very easy plant to identify that is very much present in New Jersey. So keep your eye out for the wintergreen rust. And then finally, we have the populus rust. So this one is growing on Populus tremuloides, but it supposedly will also grow on Populus deltoides, which is pretty common in our riparian zones in this area. And this will present as sort of an orange dusting on the underside of the populus leaves. This is on my list of rusts that I would like to find in the near future. Here's a nice little resource page. Special thanks to my friends who have let me use their photos: Kevin Popowich, Bethany Teigen, Stephen Berge, George Pushkal, and anyone who puts their photos on iNaturalist as Creative Commons open. Without the photos from iNaturalist, I could never have been here with you tonight presenting on this topic. So I'm very grateful to anyone who's using iNaturalist, especially if you are open to your photos being used. The two books that I use the most for identifying rust fungi are these two here, the Westcott's Plant Disease Handbook. I have an older edition of this book. I have the 6th edition. We're now on the 8th. But the 8th edition is really expensive and the 6th edition is doing me well. So I would love to encourage you to pick up this book. It is an intimidating wall of text, but it is very easy to use once you get the hang of it. And then I have another book called The Illustrated Genera of Rust Fungi, which provides a lot of context and background information about how these fungi are related to each other and some of the inner workings of the spore structures, and how to determine which genus you might be looking at. So this is another really great resource. If you are interested in the biology, the genetics, the family trees of rust fungi, there's a really amazing talk that was sponsored by the Illinois Mycological Society on YouTube by Dr. Aim out of Purdue University. That talk is amazing. I've watched it no less than 5 times and I'm still learning things from it. So if you thought that this was interesting, definitely watch that talk by Dr. Aim. She's an expert. I am simply a person who's very much in love with rust fungi and happy to be here with you tonight. So with that, I would love to move on to questions if we have any. I'm going to stop sharing my screen now.
Great. Thank you very much, Sam. That was awesome. So we do have some questions in the chat. Do you want to repeat your questions for us, Brandon?
Thank you.
Yeah, sure. So I think I spelled that right. Euromyces, the one that presents on a plant in the wetlands. What was that growing on?
That was growing on Peltandra virginica, which is a very common native wetland plant in this area. I think the common name for that plant is arrowhead.
Okay, cool. Yeah, I'm not great with plants, which makes me not great with rusts, but I do spend a lot of time in wetlands in New Jersey, so I'll keep an eye out there. Um, and then that Vankia, I think Vankia species that you showed, was it just that little black dot?
So yeah, it presents as sort of like a bubbling in the leaf of the trout lily, and then as the trout lily is beginning to degrade for the season, because it is an ephemeral plant, it will sort of split and open up, and then you'll start to see, like, it will be open and you'll see that black coating that I showed in the background of that slide. In that image, we had torn it open, so the spores may not have been totally mature at that time, but yeah, that's how it presents. It's just like some bubbling.
Yeah, so the bubbling, and then if you're so inclined to kind of split it yourself, you can kind of see the black fruiting body, so to speak, right?
Yeah.
Cool. My other question was the endocrinium, crenartium. Did that look like multiclavula? Like, were those little finger-like structures?
Yeah, that it totally looks like multiclavula. The difference here is going to be all habitat.
Mhm.
So multiclavula typically grows on really wet wood with a dusty, like bluish lichen at the bottom because it is a lichenized fungus. And usually we won't see that mixed in with mosses very much. At least I haven't observed it mixed in with mosses very much. Whereas this endocrinium is going to be growing directly out of the fronds of mosses.
Cool. Yeah, I just thought that the similarities between the two and the presence of like an algae or a moss, that's pretty interesting that they have that similar look to them. And yeah, obviously the lichenized fungus on the multiclavula, it's interesting.
Yeah. While I was creating this presentation, I found on iNaturalist a species of rust fungus that actually grows on lichens.
Oh,
And I can't remember the name of it right now. It's a boreal species though, so it would grow much more north of here. But if I ever make a trip to Canada, that's going to be on my list while I'm there.
yeah. Neat. Yeah, really cool presentation. Thank you so much.
Thank you.
And
I have
then
one more question from the chat from Lila. Are all of these rusts visible to the naked eye, or do some of them require a loop to see them? That is a good question. All of the things that we covered in this presentation are visible to the naked eye, but are seen much better, definitely through a loop. Some of them are just like a millimeter in size. For instance, during the City Nature Challenge this year, I did photograph a Jack in the Pulpit rust, but it was at the very early stages. And so I did actually need my loop to confirm that that was what I was looking at. But most of these species will be visible with the naked eye. At least you can see that there is an orange rust growing there, and then take a closer look with the loop. That's what I would recommend.
Thank you.
Another question in the chat from Hunter: are all galls caused by rusts? I'd love to see how that works again. No, not all galls are caused by rusts. Although there are many species of rusts that do cause galls. Most of the galls that I find are actually caused by insects and arachnids, who will like take their ovipositor and deposit their eggs inside of a plant, and then that plant will create specialized tissue to house those insects or arachnids. And that's the most common kind of gall that I see out there. Those are also extremely interesting and usually a sign of coevolution between the plant and the insect as well. So definitely still really interesting and something that I am for sure looking at out there. But most galls, I would say, are not caused by rust fungi.
But the one you mentioned, can you just like mention what you said about the galls in your presentation? At one point you did talk about.
Sure. Let me go back to my kitchen so I can pull up my slide again.
Thank you.
No problem. Yeah, I'll share my screen again and then I will. Can you guys see this?
It's coming up. Now we can see it.
Okay. Can you see the cedar apple rust slide?
We're at the Star of Bethlehem right now. Now the blackberry rust. Now the blue stem rust. Now we can see it.
Okay, cool. So this was the only rust that I talked about in this presentation that forms a gall. So the gall is actually inside of this mass of epithelial horns here in this like orange octopus ball. The gall is inside of that. And actually this is a perennial rust. So this will actually produce epithelial horns on the same gall year after year. But during the initial infection, when we go from the achiospores to the gall on the juniper, that the juniper tree will take 2 entire years to form that gall before it's ready to produce the epithelial horns, those like orange structures that grow out of it.
Thank you so much. I needed clarity.
Does that answer your question, Hunter?
It does. Thank you so much.
And during the little video,
Okay, great.
During the video when the little rust spore attaches to the wheat stem and just throws a little thread in there, is that a common modality among rusts?
Yeah. So in the video we were watching the spore effectively germinating. So the thread that I put in there was hyphal cells growing out into the plant. That video is really great, and I would definitely recommend watching that video in full with the original narration, because they do a really fantastic job with explaining the life cycles of rust fungi. And I will actually put the link to that video in the chat for you guys so that you can watch it in all of its glory.
Thank you so much.
Do you guys have any other questions for me? If there's no other questions, I would love to just make another couple book recommendations. When I originally gave this presentation, it was for the Philadelphia Botanical Club. So it had more of a plant focus at that time, and it was kind of expected that the audience had some background knowledge of the plants in our area. If you are new to plant identification, I would love to recommend a few books for you. We have Oh, we can't see it. So I have Wildflowers of the Atlantic Southeast, by Laura Cotterman and Damon Wyatt. It is published by Timber Press. This is the best wildflower guide for our area. This will be your best to herbaceous plants in New Jersey. No, it's Atlantic Southeast, but it does include Southeast Pennsylvania and most of New Jersey in the area that this book covers. So this would be a great place to start. And then I always recommend the Philadelphia Trees book as my favorite field guide to trees, although this crowd is not in Philadelphia. So I would love to recommend the Sibley's Guide to Trees. This is another really good comprehensive book for our area if you're looking to learn some things about woody plants. One more book recommendation is a series by Eastman.
Can you repeat that?
Yeah, another book recommendation that I have is a whole series by John Eastman. The first one is called Forest and Thicket. The second one is called Swamp and Bog, and the third one is called Field and Roadside. And these books are effectively deep dives into ecology based around a plant species. So John Eastman will describe some things about the plant and then do a deep dive into all of the organisms that are associated with that plant. And he does cover a lot of fungi. He covers a lot of birds and a lot of insects in these books as well. So if you're interested in more of like an overall ecology, this series has been absolutely amazing. It was one of the first books that I picked up in my adult life that I thought, wow, I have to read this book cover to cover. Not that I wasn't reading before, but I did a lot of like nonfiction reading in which I would read small passages out of the book. So if you're looking for a cover to cover read, that's my biggest recommendation.
Great.
And I think that's it from me.
Yeah.
Thank you so much. It's really great to have you present to us and share all this rich information and provide these resources so we can build on it.
Yeah, that was really awesome. And it's fun to have this new list of plants to look at for us now.
Yay!
Yeah, it's like now I can go on a little scavenger hunt and try to find them. I'm excited.
Yeah, I think it's one of the fun things about rust fungi for me is that it's really easy for me to choose a target species and find it, which I really like.
Yeah, I like that idea, and yeah, you set it up nicely for us.
Thank you. Oh, I got another question in the chat from Dave. Is the best time to look for rusts on plants early spring? I'm gonna say, but a lot of the things that I covered in this presentation are rusts that grow in the springtime. And the reason for that is because that is the time when there's the least amount of other fungi that I might be looking at. So if like when the weather first gets warm in March and April, I, am out there hiking and sort of jonesing for my dopamine that normally comes from the fungi that I'm observing out in the wild. But there are remarkably few species that are out that early. Rust fungi is kind of the one that is there, or the one group that is there. And they are not always on the spring plants. That's just when I'm most likely to have seen them, is in the spring. But there are a lot of them that are out in the fall too, particularly anything that is growing on an aster. That's a fall flowering plant usually, or anything in Asteraceae, so that's the goldenrods, and all of the asters. So that's a good time to look for rust as well. It would be in the fall. I think like many things, there is a somewhat of a summer slump where we see sort of a drop off. But I imagine if we have another wet summer like we did last year, we'll continue to see high levels of diversity even through the summer months.
Does this mean you're more likely to find rust in wet areas?
Not necessarily. I think that, Mike, I think you were at one of our forays last year in May, at Nakamixon, where we hadn't had any rain at all in like a month, and rust fungi were the only things that we found that day. That day we were not the Philadelphia Mycology Club. We were the Philadelphia Plant Pathology Club that day, because there were no mushrooms, but there were still rust fungi, and that is another thing that I love about them. They are persistent and they are always around. Don't, I think, particularly need high levels of moisture like other fungi would to fruit, although it certainly helps. I think they get a lot of their moisture from their plant host, which is to say that if we were having a really severe drought, they may not be able to get that moisture from their plant host at that time if their plant host wasn't thriving. And if they tried to, they may end up killing their host, which would be a bad strategy.
Very cool.
So part of the reason I asked was when we were at the foray Sunday at the Willows, none of the May apples I saw had any allodosis on it. But I had seen it earlier in the Skunk Hollow, which is the lower elevation part, which is wetter.
Mm-hmm. I think that I also noticed that because I was looking for that as a talking point myself on Sunday. But I do also think that we're now at this stage, at the time of year where the mayapple rust is transitioning from their axial stage over to their teleal stage, and they are a bit more inconspicuous in the teleal stage. They're not as like bright orange on the bottom. So it could be that we maybe missed them, although I'm not convinced it was
But
that.
it shows up as sort of faded yellow dots on the upper side of the leaf and black dots on the underside. And I didn't see any of that.
I also didn't notice any, so I wouldn't venture at this time to guess why that would have been. Perhaps all of the May apples that did have rust already got chomped on by the deer. That's what I got. I don't know. Easier to see, easier to eat, extra protein. I don't know.
Thanks.
Yeah, and I just have one more question there, Sam. Hold on, let me get back to it. Sorry, I had it written down. I lost it.
That's okay.
Oh yeah, it had to do with the jumping of hosts that you were kind of concerned with the multiflora.
Yeah.
So obviously that's a concern because the multiflora or our native roses haven't had the opportunity to kind of evolve with the rust. So there would be kind of more of a risk of them not necessarily parasitizing, but causing some problems with a new non-native host, if that makes sense.
Yeah. That definitely is what where my concern lies. I mentioned at the beginning that most rust fungi do not kill their host species that they are highly evolved with. But as we've seen with other fungal diseases like the chestnut blight or the Dutch elm disease, bringing in a new pathogen to an area where there are closely related host species, we could potentially see this becoming a serious problem for our native rose species. And I will say that like multiflora rose has been here for a really long time.
Mhm.
Even without documentation, I'm sure that the rust has been here for a really long time as well. So I'm not necessarily concerned about this particular species. I'm more using this as an example for why we should be concerned about the effects of globalization and the way that rust fungi might become a problem in that case. I hope
Right.
that makes sense as an answer.
Yeah. I mean, if the host is being introduced here, and the fungus, the rust is jumping hosts, they haven't had
Mhm.
the opportunity to evolve together, and the plant hasn't had, if at any point necessarily, built up, the symbiosis or the immunity to the rust. So yeah, interesting, cool.
Any other questions?
Looks
All right.
like you've answered all our questions, Sam.
Great. Well, thank you guys for having me. This was a lot of fun.
Thanks again for the great presentation.
Of course.
Yeah, thank you, Sam.
So we have a little more time here, and I was thinking, since we have a little more time, I'd be glad to share some observations that people found in the past week or weeks, and we could talk about them. So let's see. One that's particularly relevant is one that Hunter found. Let me share my screen. Is that okay if I share it, Hunter?
Absolutely.
All right, let's do that. Oh, just a minute. I need to change a setting on my computer.
Sam, are you still there? Oh, maybe
I
not.
am still here.
Oh, okay. Those favorite pictures of yours that you showed, like at the very beginning, they weren't yours, but the one was, they like they looked like paintings. They were so beautiful. One was the Spring Beauty rust on the right, but then the one on the left was, I think it was just the blackberry rust, but the way they were, I don't know, presented, it just looks so neat to me. I just wanted to say, I don't know what sort of additional doctoring went on, but it really looked cool.
Thank you. I do have like a pretty distinct style when it comes to my graphic design work. And I love to remove the background on different images and then have one particular specimen be like the star of the show.
Got it.
So if you have seen like our my club's business cards or like our banner on Facebook, it's a wonderful collage that I made of all different species that had the backgrounds removed. And yeah, but the Spring Beauty photo I think is like my favorite photo ever, and that's Tom Bigelow's photo from the New York Mycological Society.
Cool. So yeah, so that's thank you for sharing that. Yeah, it sounds like a fun thing to play with, and just by doing that, it takes on a whole new sort of look. Yeah, very cool.
So in order for me to share my screen, I need to quit Zoom and come back in. So Lila will be the temporary host before I return. See you in a minute.
So Sam, are you just using Photoshop to do that, or what do you like to use?
I actually use a free web program called Canva. And it is so awesome. It's really popular among people who do like content creation, social media work. But it is I have a paid version through the club because we're a nonprofit. Nonprofits get Canva for free. But yeah.
Cool.
But there is a free version that's also really good.
Thanks.
Sam, I just put another question in the chat.
Sure. Any recommendations for doing micro with Rust? Yeah. I have done a fairly significant amount of microscopy work with rust fungi. I will say I am not great at it. But I have been told by Nicholas Schwab, who is I guess the leading expert on rust, as far as I can tell, at least in like the community science world. He's really up on top of identifying things on INAT, which I appreciate. He said that looking at the axial stages is one of the important things to look at. So basically what I have done in the past is I've taken a cross section of the spermagonia where the axial spores are produced, and looking at the structure of the spermagonia, the little like sac that it grows in, and looking at the structure of the spores and how they are sort of arranged within that pustule, that is what you primarily would want to be looking at. I will say I struggle a lot with getting a good cross section of the spermagonia, just because cutting tiny little pieces of a leaf is really hard to do. Yeah, so I wish you luck on that endeavor. That is where I would go for identification. It would be the axial stage. But that book that I recommended, Illustrated Genera of Rust Fungi, that has a lot of really good images of some of the microscopic features of individual rust fungi, and sort of what you would want to be looking at for them.
Do you is most of the work done at 1,000 times magnification or maybe 400 times? Do you know?
I hardly ever use 1,000 times. 400 times is probably where I'm looking, but maybe even less than that. A lot of the spores for rust fungi are really large. Uridinospores, you can actually, and teleospores, you can actually see them like with your naked eye sometimes. They're really large.
So maybe even a stereoscope could be of some use.
I usually use my stereoscope for creating my slides, because that will help me get like the cleanest cuts. But yeah, I mean, I definitely would start by looking under a stereoscope. You can definitely see uridinospores like individual urogeniospores on a lot of these species with a stereoscope.
Okay, thank you.
Yeah, no problem.
Yeah, that's really good news. To Dave's question, don't have to go to 1,000.
Yeah, you don't have to use oil.
Yay.
I hate using the immersion oil. It gives me a lot of anxiety. I had my first microscope. We actually I guess didn't clean the oil off well enough and totally just ruined the whole thing, which is devastating. So I've been fearful of it ever since. But yeah, you definitely don't have to go that far for rust fungi.
Sounds good. There's on my property, most of the plants you mentioned can be found on my property every spring. So this gives me something new to look for April and May.
Yeah, and that the hepatica winter's over, so we could probably see that year round, I would think.
I didn't include the graphic in this presentation, but in the email that Hannah sent me regarding the hepatica rust, it included a chart of the range, the anticipated range for that species, and none of the deformed leaves had been recorded in New Jersey. So if you do find those deformed hepatica leaves, that's sort of groundbreaking information on this species. And definitely post it on iNaturalist.
Yeah, that would be a state record, right? The first time a thing is found in New Jersey. So go look for them.
As far as I know, I don't know what is in the historic collections in New Jersey. But as far as I know, it has not been posted on any community science platforms like Mushroom Observer or iNaturalist. So it would certainly be an update on some information if you did find it, which hopefully you'll be out there looking for it. I've been looking for it since very early spring when I first started to go outside again, basically. So I'm hoping that somebody nearby will find it soon.
And Sam, on that comment, do you check like Index Fungorum for these records and things like that? Are they even recorded on there as fungi? The early ones?
Oh, this is a really good question. I'll turn my video back on for this. So I have been using MycoPortal to check some of these historic records. However, MycoPortal involves somebody having needed to upload things and digitize the records, which I have been finding has not really been done at a lot of the herbaria. And so my club is currently forming a partnership with the Philadelphia Academy of Natural Sciences. At the Academy of Natural Sciences, they have the historic collections from Schweinitz, which luckily the Schweinitz collection has been well documented on MycoPortal at this time. But many of the other historic specimens have not been uploaded to MycoPortal. And so there are huge gaps in our historic data right now. And I hopefully will be working to actually digitize some of those records myself. Within a year, I'm hoping to start that project with the Academy. But I would assume that that is the same case for wherever the New Jersey records are held, which may very well be the Academy of Natural Sciences in Philadelphia, but it also might be the New York Botanical Garden.
Rutgers mostly.
Okay. I would love to encourage you guys to reach out to your local herbaria and in contact with them. And I know that the Academy was really receptive to working with our team of volunteers to this collection sort of in the 21st century. And I would have to imagine that Rutgers would be the same. Yeah, definitely take a look to see what is there. And if you're thinking that there's holes in your data, there probably is.
Yeah, that makes perfect sense, especially for things like this. The rust fungi that people haven't paid attention to until very recently. I would assume, outside of the select few.
Do you know if there's any sort of scene as far as people posting like more agricultural rusts and stuff on iNet? I don't even know why I didn't think to do it, but I had a cool one on some of my sorghum in my garden last year.
Wow, cool. I mean, I'm kind of posting anything and everything to iNaturalist. I pulled a tick off of my scalp yesterday and I put it on iNaturalist immediately.
Oh, you're a hero.
So yeah, I mean, I think you should definitely post anything that you didn't personally cultivate on iNaturalist. And that would definitely include the rust fungi. I will say, kind of going back on whether or not rust fungi were historically important, in Dr. Ames' presentation, she actually talks about rust fungi being like among the first, specifically the wheat rust, being among the first documented fungi. They have like tablets, like Sanskrit tablets depicting wheat rust, and that being like a very important both like spiritual and agricultural problem in society like thousands and thousands of years ago. But some of these more obscure things, like the little tiny dots on the hepaticas, like those may be lost historically. Hopefully they're not, though. And I'm going to be finding that out soon, and I hope that you guys will as well.
Well, what wasn't potato blight a rust?
I'm not sure.
This was a good a good presentation. And I feel like I got homework.
I love homework. If I can give you the homework, it's to watch Dr. Ames talk. Her talk is really good and gets really into like the history and the genetics, because she is actually the person who's doing a lot of the work on the phylogenetic trees of rust fungi. So definitely watch that one. Wow, awesome update from Live about the NJMA collections. Wow, and lots of rust in the collection as well. That's really cool. Yeah, I think a lot of the collections at the Academy are also going to be rust, and I'm gonna have probably a pretty good time looking through those. Probably less of a fun time looking through the Agaricales, which is probably the opposite of how most people would feel. But what can I say?
Yeah, at that point, you're just putting, some dehydrated powders under the slide, right?
I think that for us, that process is going to look more like assuming at this time that the original name is put on the specimen is correct, and uploading things as they are, and then maybe doing some backlogs to after we've sort of gathered what's in the collection, because there's like thousands of specimens that are not on MycoPortal. Sort of gathering the information and then going back to say, like, are we really sure that that was Tranchalia pernispinosae in that collection? Maybe we need to go back and do the micro. Or Hannah is hoping that by the time we get through the collection, we will have we will have a better understanding as a community on what we need to do to DNA sequence some historical specimens, which is challenging to do at this time, as far as I understand. Not that I do any of that myself.
Yeah, but those historical, I mean, they're coming from herbariums, I assume. So like a rust that would present itself on a leaf, is that leaf in there? The whole leaf?
Yeah.
Kind of. Yeah. Okay. Cool. And are the fruiting bodies, do they hold well over the years? Like, can you see the fruiting body on the leaf or no?
That's going to be specimen by specimen. Some of the things when I went for my initial visit to see the collections, some of them were completely unrecognizable as fungi. So I was like handed like a stick that visibly had
Right.
like some moss also growing on it, but I would never have recognized that as a fungus,
Mhm.
especially not a fungus of interest, from the specimen that I was looking at. And so I do anticipate that there will be plenty of challenges along the way with this project. And some of those challenges are going to come from, like, some of the specimens at the Academy are collections that were made in the Philippines. And I don't know anything about the fungi of the Philippines.
Right.
I know
Right.
about the fungi here, and really just about some of them, not even
Yeah,
all of
like
them. So
what you've learned, like barely what's actually probably out there that people have found in the past 200 years here.
yeah, for
So-
sure.
So I've got a relevant observation that Hunter made that I'd like to share. Let me do that.
Oh, wow, I've never seen this one.
So this is Phragmidium ruby IDA.
Where's it from?
Maybe. It's from Iowa, near the Mississippi. But I wasn't sure if those are red raspberry or these little, like, dwarf berries, these little tiny black, they're like black raspberries, but they're smaller, that we have so many of. So I'm not positive. But anticipating this program, I noticed a few different rusts out there. And I'm really excited now, as I mentioned, to find more.
So this, like the painting at the bottom of these leaves, does look very Gymnoconia to me. And if you are thinking that the plant looked like a black raspberry but smaller, I think we might be looking at a dewberry.
That's it. Yeah, maybe, perhaps.
So I'm gonna see if I can find dewberry in the Westcott's guide and see what we come up with here. Yep, I got it. I got dewberry. So I do have, you said this was in Iowa?
Yes.
Okay. So according to this book, which again is an outdated version, one of the species that would grow in Iowa would be Conchelia nitens, which I think did get synonymized with Gymnoconia.
Okay.
Could you put that in the chat for us?
Sure.
Mike, can you zoom in on that stem in the upper left? Is that trying to rule out whether it's the black raspberry with the kind of purplish?
I think that's a stem of something else, maybe.
Yeah.
No,
And I was gonna say that those leaves, they look a little sharp for dewberry. Like I'm thinking maybe just regular like blackberry.
Yeah, it's not a blackberry stem.
Yeah, the blackberries that we have here are usually 5-leaved. These do appear to be 3-leaved. But there very well may be lots of different species of rubus in Iowa that are not here.
That could be true, yes. Good find, Hunter.
Thank you, Lila. Yep, I'm doing my homework and now we have more.
I have been
And-
since the City Nature Challenge, I have been just like keying out plant pathogens on my couch like every day. And this book now is just like it sleeps next to me every night.
so Hunter had another couple observations. Let me
Awesome.
see if I can show those. Just a second here. Let me move this out of the way.
Oh, yeah, right. So this is a displaced Mutinus elegans, and I took a photo, like literally it was minutes later. Like I wrote the times down in iNaturalist, but it's like 20 minutes later, something like that. And it's I just think it's so cool. This patch of stinkhorns I photographed before other years, and I have this one photograph with like 6 stinkhorn eggs hanging from the base of one of the fruiting bodies. And actually, I think Arlene and Alan Bissett are going to use it in an upcoming book on the larger group that the gastromycetes, which is pretty exciting. But these are from the same patch a year later. It's where old large maple came down, and so it's a wood chip circle. But I moved the specimen because I like the mossy green background, and I wanted to get in the shade, but I find them very interesting.
Cool.
Didn't know we had a published photographer in the group. Well,
Not yet, but
adding one.
Well,
Adding one. I think there might be a few, but
there probably are, right? Yeah.
congrats. That's awesome.
Oh, hopefully, yeah. It's exciting. I can't wait for the book to come out. So I don't know when it will, but yeah, it'll be neat to have something. And then there are actually maybe more than one photo too, a couple others, which is a cool spiny puffball photos that I took. So it's exciting. And and this is not a rust either, but I found some more deer vomit in Iowa, and we all thought that was kind of interesting last week. So I was like, oh, there it is again, and it's on the grapevine again. So that was cool, and I got some close-ups. It's kind of interesting.
Wow, on grapevine.
Yeah, grapevine, right. Really weird.
I've seen
Yeah.
the species a handful of times, but every single time it's been on uh, sweet birch.
Oh, okay. Yeah, and we don't really even have much of that around. So, but not that many grapevines either. They're kind of these like lonely vines here and there. I mean that I've seen this on. So kind of interesting. And I had uploaded, but I didn't share a gall that I found in my yard today on a hackberry leaf. And I was thinking this is a rust. And and then once I posted it on iNaturalist, they said it was a gall. And so that's where my question, Kate, was stemming from, because it was so interesting looking, I thought, and it was so hard. It looked like it was like a a little, um maybe like where a spider had wrapped up a another insect in its web. It looked like these little cottony substances on the leaf that were protruding, but then when I touched them, they were hard as a rock. And so that's why I was curious, but that's just more probably what you were saying, the insect related, um, kind of the leaf warding off the insect. But it's not
Well,
here.
thank you, Hunter.
Yeah, thanks.
Cool.
Thanks for letting me share. And this was such a great presentation tonight. So good.
So are you still with us, Virginia? Oh, this is Dave's. So Dave,
Yeah, I'll be quick. This is one of the few mycenas that I can identify by site generally and by habitat or by ecological community. That I think I'm using that term correctly. Mycena praelonga grows in sphagnum moss or other kinds of moss. It has a really long stem, so you don't see the whole thing until you dig it out. It's a dark capped mycena. The spores are generally under 10 microns long, but not real small. As usual, amyloid spores, but not strongly amyloid. And it's been some discussion. I've had some discussion with Brandon about viewing spores with using a stain. Funny thing about mycena spores, they're amyloid, but if you mount them only in melters, they can be a little bit hard to see because apparently the walls are very thin. And even though when they're en masse and you put the melters on a spore drop, usually you can see the darkening right on the print. But I add some Congo red to my mount with melters, and the two of those substances together seem to bring out at least make the spores easier to photograph for one through my microscope, and then I can get maybe a little bit better read on the dimensions. That's Mycena praelonga. I think I'm saying that correctly. I find
So
it every
Dave,
spring in the same area.
so Dave, what you're saying is you mount them in KOH and then hit them with a touch of Congo red.
First with melters. See, this picture right here is a spore drop on a slide, and I added a little bit of melters, and you can see the
Oh
darkening
yeah.
sort of along the perimeter of where the melters is spreading out. That's where you'll see the amyloid reaction most readily. This particular collection did not seem to have had spores that did not seem to be strongly amyloid, but the print did darken. But then I add a little bit of Congo red, right to the melters really, and together they seem to darken the spores enough that I can get decent photos. And that's as usual, like I explained before, I work with the photos on my laptop. I blow them up and try to analyze like that. Um,
Cool. Thank you.
Mike, do you have the tricholoma there? I sent you a,
Yeah, give me a
that's a
second.
really interesting thing to find at this time of the year.
All right, just a minute. Let's
It's
see if I
gray
can.
pine associating tricoloma mushroom. I mean, it looks sort of like it could be Pterium or something close to Tricholomaterium. The gills are not gray though, so I don't think Pterium. But there's a number of these gray-capped tricholomas that grow with pine trees. Usually, you don't find them until October. So this was a really interesting thing to find. I haven't found any other mycorrhizals yet, except maybe for Morchella, but they're mycorrhizal in a very strange sort of way. The fungus produces fruit bodies triggered by nutrient deprivation. Most mycorrhizal mushrooms or mycorrhizal fungi, ectomycorrhizal fungi, would produce mushrooms when the tree has settled down and is producing nutrients that it can share. So that's like the opposite of what Morchella does. But yeah, these spores are mounted in Congo red. Oh no,
Dave,
I'm sorry. This was in a cotton blue. I've been having better luck sometimes mounting pale spores in cotton blue.
Sorry, Dave, have you blasted any of these things?
Well, first I'd have to get this thing sequenced. So,
Yeah.
it's dried. I have it. I'll look for some sort of outlet, to be able to eventually get it sequenced. But I do have the material here. It was an interesting find. Yeah, I would certainly love to have this sequenced and find out what it is. I mean, in general, even the ones you typically find in fall, the gray cap pine associating tricholomas are, I'm not sure that they're all that well understood, really.
Well, I asked that question because I know the Burkharts from the Pine Barrens have sequenced, morphologically looking different gray capped kind of scaly tricholomas, and they all come back as griseous, right?
I'm not sure I know that species. Tricholoma griseus? Yeah, you know what? I think that's one that I looked at when I was trying to, maybe formulate a proposal for this thing. Well, look at griseus. There's another one. There's a couple of them that have like scaly caps, like this one. The ones I looked at, I think the spore dimensions were sort of, maybe enough different that it seemed maybe wrong.
Yeah. I'm not questioning your ID. I'm just it's
I know.
it's more of an anecdote about the things that we keep finding in the Pine Barrens, and they all look so much different. And the DNA keeps coming back as the same thing.
Well, that might have something to do with the Pine Barrens being a rather it lacks sort of tree diversity, right?
Right.
You've got
Yep.
the scrubby pines and the scrubby oaks, and that's mostly what you have there.
Pitch pine, blackjack oak. Yep.
Mm-hmm.
So Virginia has one that I'd like to share. Let me see if I can fire that one up.
Okay. This is the less interesting one. I don't have spores. I had two that I
Oh,
sent.
let me get the other one then. Hold
Yeah.
on.
Yeah. Okay, this is in our lawn. So little brown mushrooms, probably about an inch across. Some of them were not as dark colored, some were. Okay, so that's what it looks like underneath. Brown gills.
You have a proposal for it?
No, I know nothing about mushrooms. That's cut in half. That's where I
Anybody
cut.
else want to take a stab at this one? I think I know what it is.
I'm sorry, I can't.
Paniolus?
Yeah, Paniolus phoenisecii.
Oh, okay.
Yeah,
Yeah,
I'm pretty sure that's what that is.
Yeah.
They come out on lawns fairly
Yeah.
once the
And-
weather warms up.
Oh,
Oh, you got
yeah.
spores.
Oh,
Yeah.
a lot of
Okay.
spores. That's before, I mean, when I just cut it, I dropped a lot of spores.
Yeah, they drop a lot of spores usually, but what I'm saying is, this should seal the deal then. Did you look? Let me look really quickly. Um, these are very warty. I know that Phoenicecii has really warty spores. I'm not
Yeah,
sure if they have
it-
this big Q. This would be a big Q for Panaeolus spores, meaning that they're a lot longer than they are wide.
yeah, but they have that kind of, what do you call it? A plage
The snout. No, they have a snout. Yeah,
snout.
it's a germ pore that's got like a neck on it, sort of. Um,
Yeah, you can see it in the one at like.
oh, I see. They have like a little flattened. I don't think that's a plage. I'm not sure. I really don't know. But you see that these are really warty. You can see the warts. Now I know that Panaeolus Phoenicecii has spores that are very warty. Let me see. You know what? I think I have a book right here. Let me get a book. This mushroom is commonly described in books. But I'm pretty sure that's what you have here.
Yeah.
And
I think,
what color was the print? Was it pure black or was there a sort of a
oh,
little bit of a purplishness?
spore print was like a dark brown.
Yes, Panaeolus Phoenicecii. Right. Other Panaeolus species have jet black spore
Jet
prints.
black, right?
Mm-hmm.
There's
And
sort
this
of
is in water, I assume, giving it that kind
that's
of.
in water. Yeah, that's about at 1,000. And that was the what do we call those? Cystidia, whatever they are.
Mm-hmm. Yeah.
Cystidia.
Yeah.
Looks like it to me.
Uh-huh.
And I think Foseki, whatever it is, Fosekienii has uh, highline cystidia, if I'm not mistaken, which that would make sense.
Hyaline, yeah.
Kind of
Yeah,
see
I
through.
haven't looked at them. And I should because they're on my lawn every spring. They'll be on my lawn in a few days because it's hot
Yeah,
now.
yeah.
And next time it
When I
rains.
walked along the street, everybody's yard seemed to have
Yep,
similar
Yeah.
looking ones.
Yeah.
they're in my apartment complexes. I haven't picked one up yet because I've been busy with other things, but they're out there, you know.
I'm looking
Until-
in Bironi's book, and he's got the spores for Panaeolus Phoenicecii as 12 to 17 by 7 to 9. So not quite a Q of not quite 2, but like 1.8 probably, 1.9 maybe.
That looks about right.
Yeah, that looks about right. These might be a little closer to 2. But that's what these are.
Yeah.
That's
And
what these are.
here's the spore print.
Yeah, not jet black. Yeah, just really
No.
dark, brownish, purplish
Dark
sort of.
brown.
Yeah.
Okay. Well, thank you, everyone.
Thanks, everyone.
And thanks especially to Sam for your great presentation. What are we doing next week, Brendan?
Give me 1 second.
Is Bruce presenting?
Looks for one thing. We're going to do Bruce's presentation on the 30th.
Uh-huh.
So it's a Sunday presentation.
Okay.
Yay.
Yeah. So
Or Thursday,
sorry.
I mean, Thursday.
Yeah, that's Thursday, right?
Yeah. So sorry, I was walking back inside. Next week we are going to do member finds, just our normal Taxonomy Tuesday member finds. New Jersey has been producing some good mushrooms. I think surrounding states have been as well. So we'll get back into it and just look at member finds.
Okay, Cool.
Thank you all for having me.
We
It
look
was
Yeah,
forward
Sam.
to
Sam.
cooperating more.
Yeah, I hope so. Please be in touch.
Yeah, we will. So have a good night, everyone. We'll see you next Tuesday, same time, same place.
Hey, Mike.
Yeah,
Mike, if you don't want to end the meeting, I'm willing to kind of keep hosting. If anyone has anything else they want to share, if we want to keep looking at things, obviously anyone that wants to drop off can, but anyone that wants to hang around, I'm happy to share those finds with people.
Yeah, I have one more thing you might be interested in, Brandon. Mike has the link. I can put it in a chat or whatever. It's a
and Emily says
Clarinomyces.
she has a couple to share.
Oh, okay.
Yeah, and Dave, so I get those emails as well, not just a host. So I have
Oh.
the link
Oh, good.
to that thing as
Okay,
well.
great. Yeah, I
But,
have a Clarinomyces that's not marginalis.
Yeah, neat.
So I'm going to stop the recording and drop off, and I'm going to make Brendan host.
Cool.
So Lila, could you stop? I think you're in charge of the recording now. Can you stop it?
No. Okay.